Hey, welcome back. Today we are starting eukaryotic cells. Last couple of weeks, we've been looking at prokaryotic cells. So today we're starting eukaryotic cells, and we'll be looking specifically at protein pathways. So the pathway that proteins take from their instructions to their formation, to their sorting, and eventual exit from the cell, potentially. So we're looking at all the organelles involved in that process. So starting off kind of at the beginning here, the genetic code, the instructions for proteins, are contained in an organism's DNA. And if you recall, proteins are doing all the work of the cell. They're what give you your brown hair, or your blonde hair, or your black hair. They're what give you your eye color. So proteins really have a lot to do with the outcome of the cell and eventually the outcome of the entire individual. So the instructions for those proteins come from an organism's DNA, and we call that chromatin. It's a combination of DNA and proteins, and we see that here. So the DNA is the yellow strand. It's wrapped around these silver and blue and green proteins that kind of help keep it organized. Generally, chromatin is a loose spaghetti-like structure, so it looks like a bowl full of spaghetti. We see that here on the left side of your screen. The purple bowl here is the nucleus, and again, generally chromatin is kind of loose and looks disorganized to us. It's only when the cell is preparing to divide, and we'll discuss that later in the year, that the chromatin condenses into these individual strands we call chromosomes. So here's a chromosome on the right of the image. And this is usually what we think of when we think of DNA, as a chromosome. But generally, DNA is not in its chromosome form. Again, it's only around the time of cell division that this occurs. Usually, DNA is just a loose bowl full of spaghetti. So different from prokaryotes, eukaryotes have chromosomes. These are strands of DNA. They are not circular, and generally eukaryotes tend to have several of these strands. If you remember prokaryotes, they tended to only have one strand of DNA, and it was circular. Fruit flies have 8 total chromosomes. Humans have 46. But that does not mean that the level of sophistication of the organism has anything to do with the number of chromosomes. So, for example, a pigeon has 80 chromosomes. It's a lot more than us. And there's even an adder's tongue plant, which is kind of like a fern, that has 1,260 different chromosomes. Next up, we're taking a look at the nucleus, probably the most recognizable difference between prokaryotic and eukaryotic cells. You can even see the nucleus, or at least the dense portion of the nucleus, under a light microscope, which are the weaker of the microscopes, and when you look at most eukaryotic cells. So it's a large and prominent organelle. It serves as the storage and protection of the genetic material, which is your DNA, and it also acts as a control center. When we talk about the nucleus, we include the nuclear envelope, chromatin, which we just discussed, and the nucleolus, which we will discuss in a little bit. So, again, the 2 jobs the nucleus has is it stores and protects the DNA. I think it's definitely shown that it protects the DNA, because eukaryotic cells are thousands of times less likely to mutate than prokaryotic cells. So prokaryotic cells don't have the nucleus, they don't have that protection, their DNA is just kind of out in the open, subject to all sorts of environmental conditions that can potentially and do mutate the DNA. Eukaryotes don't have to worry about that as much. That nuclear envelope protects their DNA from a lot of environmental conditions. How does the nucleus act as a control center? Well, the nucleus is able to control what goes in and comes out of the nucleus. So by determining who gets to come in and read the DNA sequence and who gets to leave with the DNA sequence is the way that the nucleus acts as the control center for the cell. So the nucleus is surrounded by the nuclear envelope. Now, different than just a regular old membrane, the nuclear envelope, an envelope has two membranes. So we kind of see that here. We've got a double layer, our phospholipid bilayer. I mean, bi means two, so 2 layers of phospholipids, and then a second phospholipid bilayer. The space between is called the perinuclear space. So there's a little gap between the outer membrane and the inner membrane. So the outer membrane has ribosomes embedded into it, very much like the rough endoplasmic reticulum, which we'll discuss later. It kind of goes to show that the outer membrane is actually continuous, meaning it's connected with the rough endoplasmic reticulum. So this membrane on the outer part of the cell, of the nucleus, is the same membrane. It's continuous with the ER. So take a look at the ER, and we'll see how that membrane just continues and kind of folds out and around as part of the ER. Also included in the nuclear membrane are tons of channel proteins, which you can see them here. They're called nuclear pores, which allow things to pass through the nuclear membrane, either into or out of the nucleus. Most of these nuclear pores span both membranes, the top and the bottom membrane, the outer and the inner membrane, and so that makes them quite large proteins. The main purpose of the nuclear envelope is to act as a barrier to separate the inside of the nucleus, which is called the nucleoplasm, from the interior of the cell, which is called the cytoplasm. So also found within the nucleus is the nucleolus. See that here? It's kind of this dense red core in the middle of the nucleus. Plural is nucleoli. They are a combination of DNA, RNA, and protein. They're, again, very densely packed. And this is the site of ribosome formation. This is where the ribosomes are made. Mammalian cells, that's you and I, we have 5 to 10 million ribosomes per cell. So it's almost like a whole city of ribosomes, and the nucleolus is what makes ribosomes. Some eukaryotic cells have multiple nucleoli. So it's a very important job in making those ribosomes. So transcription and translation. So the first step in reading that DNA and building protein is called transcription. In transcription, we take our DNA strand and we copy it into an RNA strand. We see that going on up here, top left. Transcription takes place within the nucleus, so we're still inside the nucleus there. The second step of building the proteins, the RNA, so the RNA has left the nucleus. We see that right here. This yellow strand is the RNA. It leaves the nucleus. And the second step is that the RNA code is read by a ribosome and translated into a growing amino acid chain. So here's our polypeptide, or amino acid chain, which eventually will become a protein. So that process is called translation. We are translating the code from nucleotides to amino acids. And we'll talk a lot more about this process later in the year. These ribosomes may be free-floating or embedded in a membrane, such as those embedded on the rough endoplasmic reticulum. So a little closer look at ribosomes here. Again, they can either be free-floating. We see these blue circles out here are free ribosomes, just kind of floating out on their own, and they can pretty much go anywhere in the cell. Or they might be attached to the membrane of, generally, the endoplasmic reticulum, the rough endoplasmic reticulum. So that's what these are here. They're attached to the membrane. Again, here is the process of the ribosome reading the RNA strand and translating it into an amino acid strand. Again, we'll talk a lot more about this later in the year. So next up, we have the endoplasmic reticulum. This is generally the largest organelle by space in any eukaryotic cell. It is surrounded by a phospholipid bilayer. So we talked about our membrane-bound organelles. This is definitely one. And this membrane is connected, it's continuous with the outer membrane of the nuclear envelope. And we see that right up here. So purple circle is the nucleus, and if we see that outer membrane just kind of leaps out, and boom, we've got our endoplasmic reticulum kind of continuing on. It's continuous with the outer membrane of the nucleus. The inside, these folds inside of them, so kind of a darker red inside of the membrane, that area is called the lumen of the ER. So the endoplasmic reticulum has two distinct parts. It has the rough endoplasmic reticulum, we call it the rough ER, and it has the smooth endoplasmic reticulum, called the smooth ER. So a closer look at each of those. The rough endoplasmic reticulum is generally found directly surrounding the nucleus. We can kind of see that here. The yellow endoplasmic reticulum surrounding the nucleus here. And it's called rough because it has ribosomes embedded into it. So if you've ever felt Braille, if you've ever run your finger across a page with Braille, those little dots, that's very similar to what the rough ER would feel like if you could run your finger across it. The rough ER provides specific areas for ribosomes to be made. So if you think about it, you don't want your cell to be incredibly acidic, or basic for that matter, but sometimes proteins need that high acidic area or that basic area to fold the protein correctly. So the rough ER provides an area for that to happen. So as long as we keep just this particular part of the lumen very, very acidic, it's a good place to make proteins that need to fold correctly in an acidic environment. So proteins can either be made and inserted directly into the membranes. We see that here. Here's the ribosome, this white character here. The mRNA is this blue strand, and it's making a protein, this longer blue strand here. And it's possible for the ribosome to make that protein, and now it's embedded into the membrane. And that's how we get our membrane-bound proteins. Talked about those last chapter. They're made directly into the membrane. Or proteins are sometimes just made in the lumen, and then the lumen buds off into a vesicle. We see that here, and now we've got proteins inside the vesicle, and we can do a lot of different things with those proteins. The smooth endoplasmic reticulum, usually found outside the rough endoplasmic reticulum, is called smooth because it doesn't have those ribosomes. Now, the smooth endoplasmic reticulum doesn't really fit into today's lesson. It's not really a site of protein assembly, but rather it's where lipids are generally made. So if you remember hydrophobic fatty acid tails, it means they're afraid of water, and the cytoplasm is mostly water. So it wouldn't do well to just make lipids out in the open in the middle of the cell. So instead, we make lipids in the actual membrane. We just continue to add them to the membrane, and the membrane gets bigger, and then the smooth ER can kind of bud off into a vesicle and send those vesicles to other parts of the cell that need more membrane. So kind of a cool enzyme called flippase, which flips phospholipids from one side of the membrane to the other. So you see here, new phospholipids are only ever made on one side of the membrane. And so it's an enzyme called flippase, which I think is a really cool name, that flips those phospholipids to the other side. So you always have equal amounts on both sides. All right, next up we have vesicles. And vesicles are these membrane-bound sacs that contain and transport proteins. Finally now, we take a look at the Golgi apparatus. So once proteins are made at the ER, they bud off into a vesicle, and they generally travel to the Golgi apparatus. It has two sides, the cis face and the trans face. The cis face usually faces towards the nucleus, and that's the area that the vesicles enter with their proteins. The trans face is the exit end, so proteins come in, they travel through the Golgi, and they exit through the exit face. While they're in the Golgi modifies them. They add different things to different proteins, they clump them together, they make them larger, they add different proteins, they add different glyco. You see glyco there, that'd be carbohydrates, carbohydrates. So a lot of modifications can take place in the Golgi. And then finally, the Golgi sorts and sends them out on their way. That might be to lysosomes, it might be to the cell membrane, it might be out of the cell, secretion from the cell. And so that's what we're looking at today.